Okay, Assalamu Alaikum students. Today I'm going to teach you about the structure of nephron first and then adaptations of different parts of nephron for the process of urine formation. So let's start. Let me share the screen. If you want to ask any question, you can ask Uruj. No issue in that. Okay. So
Okay.
let me share the screen with you. So the process is structural and functional adaptations of nephron, structural and functional adaptations of nephron for the process of urine formation. So yesterday I told you that this is how the structure of nephron is. First of all, it has a Bowman's capsule, also called as renal capsule. And then I told you that it has, I'm not going to draw the blood capillaries, peritubular capillaries in detail, but I will be showing glomerulus. And the collecting duct. So these are the all the parts of a nephron. This is the whole nephron. And over here, as I've told you, dorsal aorta gives out branches to make up the afferent arteriole, which further gives out branches to make up efferent. In between are the capillaries called as glomerulus. There are almost, as I've told you, 50 capillaries in this. It's a cluster of capillaries. Cluster of capillaries called as glomerulus. This is afferent arteriole, bringing the blood from dorsal aorta, and this is, I made it quite thin, it's not that thin. Efferent arteriole taking the blood away, and then as I've told you, I'm not drawing it now, but as I've told you that this efferent arteriole then give out branches surrounding the nephron as peritubular capillaries. Okay, so this glomerulus along with the Bowman's capsule or the renal capsule is together called as renal corpuscle together, and ultrafiltration takes place over here. In this portion, ultrafiltration, which is the first portion, together with these arterioles and everything, what happens over here? Ultrafiltration takes place over here. We will discuss the adaptations of these parts in detail as well. Ultrafiltration takes place over here. Next is the proximal convoluted tubule. Is convoluted, and as I've told you, here are like this. This is cortex. This is medulla. Mostly the upper and the distal and the proximal convoluted tubule. Mostly they are in the. I should have drawn it. Like this, and the loop of Henle is mostly in the medulla region, and distal and proximal convoluted tubule are in the cortex region. So this is proximal convoluted tubule. This is the from where nephron filtrate goes down is the descending limb. Of nephron, and this is from where the filtrate goes up is the ascending limb. This is the ascending limb of the loop of Henle. We should specify the name, the loop of Henle. This is the ascending limb of the and then it goes from the ascending limb, it goes to the distal convoluted tubule, and then from the distal convoluted tubule it goes to the collecting duct. Which collects urine from multiple, as I've told you in the previous class, it collects urine from multiple nephrons like this. If this is one collecting duct, it collects urine from multiple nephrons. These are different nephrons, and that's the collecting duct. From each nephron, I've not drawn the nephron in so much detail, but I just want to represent that these are the nephrons, and this is how nephrons from all the directions are draining urine into the collecting duct, because this keeps on collecting and let this urine goes to the renal pelvis, and then from the pelvis to the ureter. So This is how the nephron is. Now, if we talk about nephron, let's start discussing the adaptations of each and every part of the nephron. Proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct are responsible for the process of, they are all responsible for the process of selective reabsorption and tubular secretion. So they are responsible for these. Now the adaptations of each and every part of the nephron. First of all, we're going to start with the renal corpuscle. That's also called as Malpighian body. The first part where ultrafiltration takes place. Now adaptations of the or it's also called as Malpighian body. So the Bowman capsule, cells on this side of the Bowman capsule are called as, made up of this wall of the Bowman capsule is made up of this wall of the Bowman capsule, which is not in direct contact with the glomerulus, is made up of, is called as parietal wall. And it's made up of squamous epithelial cells. Squamous epithelial cells. It's made up of squamous epithelial cells. It's the parietal wall, and it does not have any pores in between. Nothing seeps out of this side of the wall because these are squamous epithelial cells tightly packed, and there's no gap, no slits in between. But if we talk about this wall is this side wall, which is in direct contact with the glomerulus, is called as visceral wall, and it is made up of special type of cells having slits in between and having finger-like projections. That's why they are called as podocytes. Finger-like projections. And they are called as podocytes. They have slits. called as slit pores between them. There are gaps in between them. These cells are called as podocytes. Having finger-like projections and have gaps in between called slit pores. Of podocytes. So this side wall is called as visceral wall. This is called as you don't need to remember the names of visceral wall and parietal wall. You just need to remember that this side wall, which is in direct contact with the glomerulus, has pores in between called the slit pores, and they have finger-like projections to increase the surface area for maximum absorption. And this side cells are squamous epithelial cells, and they do not have pores. You just need to remember this. Now this is the glomerulus. So this is the cluster of capillaries called glomerulus. And if we further talk about glomerulus, then there is a basement membrane between the cluster of capillaries and between the visceral wall or the wall of nephron made up of podocytes. So blood comes in through the dorsal aorta and through this afferent arteriole into this glomerulus, and when it passes through the cluster of capillaries over here, the glomerulus, it seeps down and crosses three barriers. The three barriers here are endothelial cells of the capillary, which also have gaps in between, basement membrane made up of glycoproteins and collagen, and the podocytes with finger-like projections and slit pores. So these are the three barriers which blood has to cross while coming into the glomerulus. That's the capillary endothelium. First is capillary endothelium. Second is basement membrane. Third are the podocytes with slit pores. So these are the three barriers. If we talk about the first barrier, which has gaps, endothelial cells of capillaries have gaps through which the materials can seep down. have gaps. Materials seep down. Secondly, basement membrane is made up of glycoproteins and collagen, and anything smaller than 69,000 relative molecular mass can seep down, can pass down the basement membrane. Down the basement membrane easily. Third barrier is of the podocytes, which is exactly, again, just like these two, not a very good barrier because they have slit pores through which materials can seep down. They have slit pores through which materials can seep down easily. So these are the three barriers from where the blood and the materials have to cross and have to seep down into the first portion of the nephron, that is proximal convoluted tubule. Next to Bowman, this is the first portion, so it has to come to the proximal convoluted tubule. We will discuss the process of ultrafiltration in detail and the working of basement membrane endothelial cells and podocytes in the other class. Today we are doing the adaptation, so let's discuss the adaptations of proximal convoluted tubule. If you talk about the proximal convoluted tubule is made up of brush bordered cells. Like if this is the proximal convoluted tubule, it has brush bordered, or you can say ciliated. They're not like podocytes, not like fingers, they are like brush. They are made up of brush bordered cells. That is proximal convoluted tubule. So these cells are brush bordered. They do not have any gaps in between. Brush bordered or ciliated, or we can call them micro. Better name for them is microvilli. These are the microvilli actually. To increase the surface area, ratio volume for maximum absorption. Then there are no gaps. Secondly, no gaps, tight junction, no gap. Everything has to pass through the cell. Nothing can pass from between the cells because there is no gap. They're tightly packed. Then, third adaptation is the cells of Proximal convoluted tubule have a lot of mitochondria for active absorption of materials. Lot of mitochondria means a lot of ATP formation, means easy active transport. They have a lot of ribosomes. These cells have a lot of ribosomes to make up the special proteins, which are the co-transporter proteins. Ribosomes are meant for protein synthesis, and they have a lot of ribosomes to synthesize co-transporter proteins in the cell membrane, which are very important. You will understand it afterward, that what are these proteins doing, why active transport is going on. These are and they have a lot of proteins as well in their cell membrane. They have a lot of proteins in their cell membrane for the process of transport of and for the absorption of materials. So these are the adaptations of the cells of proximal convoluted tubule. We will study in detail about these adaptations, that what, how and what are these adaptations doing and helping in the process of urine formation. Next is after the adaptations of proximal convoluted tubule. Next is the adaptations of the loop of Henle. Loop of Henle, descending limb, ascending limb. Descending limb is more permeable to water and it's not permeable to ions. If there is permeability to ions, very less permeability for the ions. And ascending limb is permeable to ions and it is impermeable to water. These are the adaptations of the loop of Henle. Loop of Henle, if we talk about, it is thirdly loop of Henle. We call it loop of Henle. Descending limb and ascending limb, this is permeable to water and permeable to water and less permeable to ions. Ascending limb is permeable to ions and impermeable to water. If we talk about the collecting duct, the distal convoluted tubule, its first portion is just like the ascending limb. First portion is like ascending limb, permeable to ions and impermeable permeable to ions and impermeable to impermeable to water. And second portion is like the descending limb, permeable to water. Next is the collecting duct. If we talk about the collecting duct, 90% water is reabsorbed here, and both the cells of DCT and collecting duct are under the controlled by antidiuretic hormone. 90% water is absorbed over here and it is permeable to both water and ions. So beta, these are the adaptations of different parts of the nephron. First, I drew nephron, different parts of nephron. Then I told you that each collecting duct collects urine from different multiple nephrons. We will discuss urine formation afterwards. First, we needed to discuss the adaptations of each and every part of nephron. We have discussed that in Bowman capsule and in renal corpuscle, there are three barriers from where the materials have to pass down in the form of nephric filtrate into the proximal convoluted tubule. That is the blood capillaries having gaps, the basement membrane which allows anything less than 69,000 relative molecular mass to pass through, and the podocytes having slits. So maximum things pass down from the blood into the nephron, into the Bowman capsule and then the proximal convoluted tubule. Next is the cells of proximal convoluted tubule. They are brush bordered, they have a lot of microvilli, they have a lot of proteins, they have co-transporter and different type of proteins for the absorption of ions. Either ions are actively transported or diffuse in, or they move in. If it is water, then by osmosis water moves out of the proximal convoluted tubule back to blood. So these cells are responsible for these things. That's why they have a lot of mitochondria for ATP production. They have a lot of ribosomes for those protein, cell membrane proteins production, and they do not have gaps, tight junctions, so that everything has to cross the cell and then it has to go to the interstitial fluid surrounding the proximal convoluted tubule. Then the loop of Henle, descending limb is permeable to water, ascending limb is permeable to ions. Distal convoluted tubule, its first portion is permeable to ions like ascending limb, and second portion is permeable to water like descending limb. Then collecting duct is permeable to both. 90% of the water is reabsorbed in the collecting duct, and both collecting duct and distal convoluted tubule are under the control of the ADH hormone, which controls that how much water should be excreted out of the body and how much should be restored back into the body, depending on the conditions and depending on the water intake of a person. So any problem to you with it?
No, ma'am.
Anything which is not clear to you? You want
नहीं
to
मैम,
ask?
ठीक है.
Okay, and thank you very much. Inshallah, we'll discuss urine formation in the next class, that how along with these structural adaptations like podocytes and brush bordered cells and all these, how do they help in the process of urine formation? In the next class, we'll do it, inshallah. Okay. Allah Hafiz.
ओके, अल्लाह हाफिज़.